Hi, my name is Jennifer, and today I will be introducing the DNA binding protein p53. So what makes p53 so important? It's a tumor suppressor protein, often referred to as the guardian of the genome, because it plays a central role in preventing cancer by orchestrating cellular responses to stress. When cells encounter DNA damage or other threats to genomic stability, activated p53 acts as a transcription factor, regulating genes involved in cell cycle arrest, DNA repair, and apoptosis. By promoting these processes, p53 safeguards against the proliferation of damaged cells and the initiation of tumors. Mutations in p53 are common in many cancers, so studying it becomes crucial for devising effective cancer treatments. Now, how does p53 work at the biochemical level specifically? Firstly, p53 is a tetramer, meaning that it is composed of 4 subunits, as you can see here in the 4 differently colored shapes. Each monomer binds to a specific site on the DNA. These sites are known as quarter sites, and 2 head-to-head quarter sites you can imagine as 2 head-to-head arrows here form a half site. The directionality of the arrow corresponds to the specific DNA order sequence, but I will leave it at that for the sake of time. The p53 tetramer is split into 2 dimers, each dimer occupying a half site on each side of the double-stranded DNA. The dimer-dimer interactions increase DNA binding affinity of p53. You can think of p53 as a tetramer binding to DNA like a pair of clamps. When p53 acts as a transcription factor, it will bind to its designated site on DNA and initiate transcription. p53 responsive genes have been shown to contain p53 binding sites either upstream of the first exon or within the first intron. Now that we know how p53 binds to DNA, I'm going to introduce a specific pathway of p53, which is inducing the expression of PUMA, a gene that promotes apoptosis. Researchers have found that intron 1 of PUMA contains a p53 binding site. When the cells undergo stress, p53 is activated and binds to its binding site on PUMA. The PUMA protein is then therefore expressed, initiating a biochemical cascade that eventually leads to apoptosis. Overall, p53 has a multitude of roles in tumor formation, making it a very interesting subject to study. Understanding of the structure of p53 and its role in cancer development is essential to creating effective cancer treatments. Thank you for listening.